Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University Information Technology, Himachal Pradesh. Today I am going to tell you about Nucleocytoplasmic transport, which is present in Molecular and Cellular Biophysics paper. Nucleus and cytoplasm are two major entities which are responsible for all biological work performances of a living cell. Cellular functions are usually carried out by proteins which are synthesized by translation machinery, and it is present in cytoplasm. For protein translation, molecular signals are required from genomic DNA, which results in synthesis of different RNA. Therefore, to complete the process of protein synthesis, transport of RNA has to be carried out from nucleus to cytoplasm. Similarly, proteins from cytoplasm get transferred into nucleus. All these mentioned processes show a continuous bidirectional molecular transport between the nucleus and the cytoplasm. Present module provides information using following objectives for such nucleocytoplasmic transport and also structural elucidations of moieties which are responsible for these transport functions. We have summarized the following learning objectives to achieve the information. Number 1, to elucidate the structural components of nucleus responsible for molecular transport. Number 2, to understand the basic structure and function of nuclear export machinery. Number 3, to classify the proteins of nuclear import and elucidate their function. And lastly, number 4, to elucidate the transport of different RNA molecules from nucleus to cytoplasm. We're just coming to the introduction part of this module, in which you will find that why nuclear transport is required. If you see, nucleus is the largest cell organelle in animal cells that occupies approximately 10% of cell volume, with a volume of 6 micrometer diameter of a mammalian cell. Genomic DNA is packed inside the nucleus with nuclear and Mrna boundaries for separation of cytoplasm from nucleoplasm. However, a continuous exchange of biomolecules like proteins, RNA, nutrients, they take place through nuclear envelope, which is crucial for homeostasis between these two portions as well as survival of the cell. This exchange is carried out with the help of active nuclear cytoplasmic transport system. In this system, large cargo are recognized and bound to the soluble transport receptors that mediate transport through nuclear pore complex embedded in the nuclear envelope. This transport is highly coordinated, and any interference in transport process leads to abnormal function of the cell and severe human diseases. Therefore, understanding the role of nuclear transport machinery and types of molecule transport like proteins, RNA, etc. in cellular organization is a necessary in the cell biology. Now coming to the part of structural component of nucleocytoplasmic transport. The nucleus is crammed into two concentric membranes which are called as nuclear envelope, which are perforated by a pore complex that we call nuclear pore complexes. If you see in the figure, you will find that two membranes are separated. Even though inner and outer membrane of nucleus are continuous, they sustain a discrete molecular component. For example, proteins that acts binding site for the chromatin and provide a structural support for nuclear envelope are always present at inner nuclear membrane. Inner nuclear membrane is encircled by the outer membrane that is uninterrupted with the membrane of the endoplasmic reticulum, which are coming outside. The outer membrane of these continuous membrane is attached with ribosomes and engaged in translation product. Protein translated on these continuous membrane are transported to a perinuclear surfaces between inner and outer membrane. Continuous bidirectional trafficking of biomolecules occur between the nucleus and the cytoplasm. Nuclear proteins like histones, gene regulatory proteins, and replication and transcription enzymes are specifically imported into the nucleus from cytoplasm. Simultaneously, tRNA, mRNA, rRNA are have to export it to the cytoplasm from nucleus. Now, if you see the figure which shows the structure of nuclear envelope showing continuous inner and outer membrane of nucleus, which are perforated by nuclear pore complexes. Nuclear envelope of each mammalian cell contains approximately 3000 to 4000 nuclear pore complexes, which are responsible for molecular exchange between the nucleus and cytoplasm, and also prevention of materials not to transport which are not destined to cross the nucleus. It means they have to remain into the nucleus. The nuclear pore complex is one of the largest protein complexes of a cell with a molecular mass of approximately 125 megadaltons, or they will be present in yeast and they have 66 megadaltons. Structurally, nuclear pore complexes appear octagonal, membrane embedded structure containing 8 filaments into nucleoplasm, each about 100 nanometer long. Terminal rings join these filaments inside the nucleoplasm and form a structure called nuclear basket. Each nuclear pore complex is composed of about 30 different types of proteins called nucleoporins, and multiple copies of these proteins are arranged in prominent octagonal symmetry. A subset of nucleoporins containing phenylalanine glycine, that we call the FG repeats, is a core of 20 to 30 nanometer wide nuclear pore complex. This FG repeat rich domains forms hydrophobic meshwork that allows the diffusion of macromolecules greater than 40 kD of size. This figure shows the structure of nuclear pore complex. In every second, single nuclear pore complex can transport up to 500 molecules in both the directions. The mechanism for this coordination is still not fully understood. However, one or more aqueous passages present in each NPC or nuclear pore complex allows very fast passive diffusion of small water-soluble molecules up to 5 kD limit. Transport of macromolecules through NPC is fundamentally different from transport across the membrane of other cell organisms, where proteins are transported through a large pore instead of transporters spanning the lipid membrane. This is the reason of transport of fully folded nuclear protein inside and ribosomal subunits outside the nucleus. Proteins localized in the nucleus of an amphibian oocyte contain a stretch of amino acids near its C terminal that we call nuclear localization signals that enables protein to pass through nuclear pore complex and enter the nucleus. These NLS signals are generally present as amino acid sequence in a protein, and they are thought to loop on protein surfaces. These NLS are composed of one or two stretches of positively charged amino acids, which are basically lysine and arginine. And if you see the signals, they started from proline and they have lysine, arginine, lysine, valine. I'll give the example. NLS signal was identified on T antigen and coded by SV40 virus. If a single amino acid of this NLS sequence is replaced by neutral amino acid, that protein will unable to localize in the nucleus. On the other hand, if non-nuclear protein like serum albumin get fused with NLS signals and injected in cytosol, the protein get localized into the nucleus. Transport through NPC can be visualized by attachment of NLS with gold nanoparticle that are injected into the cytosol that can be recorded with the help of electron microscope. Transport begins when these particles bind with the tentacles like fibrils of NPC into cytoplasm, probably with the regions of NPC that acts as barrier for transport of large molecules that are pushed away, and hence coated particles can enter through. Now coming to nuclear import receptors. Localization of nuclear proteins that contain NLS is only takes place after the recognition by nuclear import receptors. Import receptors are present in the cytosol as soluble proteins and have ability to interact with both, that is to the NPC and NLS of cytoplasmic proteins. FG repeats of nucleoproteins acts as binding site for import receptors. They are thought to form a patch inside NPC for transport of cargo proteins bound to import receptors. Transport of this receptor cargo complex is not just through binding with FG repeats, but it includes binding, dissociation, and rebinding with FG repeat sequences. After transport of cargo proteins into the nucleus, nuclear import receptors get dissociated from cargo proteins and returned back to the cytosol. Direct binding of nuclear proteins and import receptor is not obvious, but sometimes additional adapter proteins can form bridges between import receptor and nuclear localization signal of nuclear protein to be transported. The another thing is nuclear export receptors. The export of large molecules like ribosomal subunits and RNA from nucleus is only possible in the presence of nuclear export signals, which are present on the surface of these macromolecules and their complementary nuclear export receptors. These export receptors are capable of binding with both to the nuclear pore complexes, proteins, and export signals. It has been found that the structure of many nuclear export receptors is similar, very similar to the nuclear import receptors, and both are encoded by same gene family of nuclear transport receptors or karyophylline. It is expected that both these transport systems work same way but in opposite direction. Coming to a specialized system that we call the Rna Gtpa system, which are required for transport through nucleopore complexes. Nuclear specific proteins are concentrated into the nucleus. Energy required to achieve this process is obtained by hydrolysis of Gtp by a protein that we call the Rna Gtpas. Rna is basically Ra related nuclear protein, is a small G nuclear protein with Gtp binding domain. This Rna is involved in both processes, that is nuclear import as well as nucleus export, and is found in nucleus as well as cytoplasm. Rna is essential for the translocation of RNA and protein through the nuclear pore complexes. Similar to other GTPas, Rna also exhibit in two conformation depending upon binding of GTP and GTP. Conversion between GTP and GDP is triggered by two RNA-specific regulatory proteins that includes a cytosolic GTPase activating factor, that's we call a GAP, and a nuclear guanosine exchange factor, that's we call GAF. This figure 3 shows a schematic representation of import and export of molecule through nuclear pore complex with the help of Ren-GEF system. Ren-GAP is accountable for hydrolysis of GTP and therefore converts Ren-GTP into Ren-GDP, where GAF guanine exchange factor is responsible for conversion of Ren-GDP into again Ren-GTP by exchanging Ren-bound GDP to GDP. As Ren-GAP is located in the cytosol, it is responsible for presence of Ren-GTP in the cytosol. Similarly, location of Ren-GEF in the nucleus is responsible for presence of Ren-GTP in the nucleus. This gradient of two Ren conformation is principal for major nuclear transport in appropriate direction. When import receptor is loaded with appropriate cargo protein, it leads to the docking of nuclear import receptor with FG repeats on cytosolic side of the NPC. Once they reach the nuclear side of the NPC, Ren-GTP bind to them and allow the release of bound cargo. Since Ren-GTP present in cytosol, it is incapable to bound cargo receptor and hence unloading cannot occur in cytosol. In this way, localization of Ren-GTP in nucleus create a directionality of transport across NPC. Now coming to a bigger figure, that we call the RNA export from nucleus to cytoplasm. If you see, for successful gene expression, different RNA species has to be transported from nucleus to cytoplasm. Small RNA like tRNA, microRNA, etc. are transported by directly binding with the export receptors. However, mRNA and ribosomal RNAs are larger RNA species which first assemble into ribonucleoprotein particle, that's called RNP particle, and then they recruited their exporter via class specific adapter proteins. Primarily, mRNA precursors undergo 5' end capping, splicing and cleavage polyadenylation at 3' end. During this process, the mRNA interact with different kind of proteins and form a stable messenger ribonucleoprotein, that's you call the mRNP particle. Now, I'll just talk about tRNA and miRNA export. Although tRNA are synthesized as a larger precursor in the nucleus by RNA polymerase III. However, these are needed in the cytoplasm in aminoacetylated tRNA form with a peculiar short length containing single-stranded loops and double-stranded mini helix regions that fold in a uniform clover leaf structures for ribosomal translation. Therefore, before export to cytoplasm, tRNA undergoes processing steps which include 5' and 3' trailer sequencing removal, addition of CCA nucleotide at 3' end to form amino acid acceptor stem, intron removal if they are present by tRNA splicing endonuclease, and base modifications by tRNA modifying enzymes. After these processing of mature tRNA are preferentially attached to a exporter receptor which is known as exportin T. It is a member of the family caryophyrin in a Ren-GTP dependent manner and exported to the cytoplasm. In cytoplasm, Ren-GAP stimulates GTP hydrolysis on Ren, which induces dissociation of tRNA from the complex of tRNA export in T, Gtp. Similar to export in T, we have another member of karyophylline family which is known as exportin 5, and it is responsible to export miRNA, and these miRNAs are regulatory non-coding RNAs. Now coming to a export of snRNA. These are spliceosomal RNA which participate in the removal of intron from pre-mRNA, and they are placed in between small tRNA, miRNA and long mRNA or rRNAs. Although snRNA are required in the splicing of mRNA in the nucleus, but their export from the nucleus and import again to function in splicing is still unknown. However, it was proposed that the cytoplasmic export of snRNA during biogenesis might provide a proofreading step to prevent nuclear accumulation of non-functional snRNA. All snRNA except U6 are synthesized by Pore 2 as pre-snRNA, and these acquire a 5' cap which has the nuclear export signal. Export of snRNA require nuclear export signal containing adapter proteins, that's called FACS, for recruitment of export receptors. CRM1 is the export receptor for snRNA which does not directly interact with the snRNA cargo but require the cap binding complex, that's called CBC, and FACS adapter proteins to be targeted to the 5' cap of the snRNA. Phosphorylation of FACS in the nucleus is required for recruitment of CRM1 and Gtpa to bind to the CBC bound snRNA complex. After export to the cytoplasm, Gtpa hydrolysis of Rn and FACS dephosphorylation takes place, which dissociate the export complex, and snRNA get released into the cytoplasm. In the cytoplasm, the survival of motor neurons, that's called SMN complex, facilitates the assembly of exported sRNA with the heteroheptameric rings of SM proteins, which bind to the conserved SM binding site of snRNA. SM proteins binding induces trimethylation at 5' cap and exonucleotic degradation of 3' trailer sequences. These processes make a pre-snRNA into mature snRNPs, which provides a composite nuclear targeting signal for subsequent nuclear import. After re-import into nucleus, snRNPs together with other splicing factors assemble into the functional spliceosomes. Now student, we will talk about the export of mRNAs. Eukaryotic mRNA are synthesized by Pore 2 as pre-mRNA and have different lengths, sequences and structures. However, mature mRNA basically share common structural features like 5' end capping, splicing and polyadenylation at 3' end. The mRNA export pathway is conserved from yeast to humans, although every model organism has its own unique properties. During the successive step of messenger RNPs formation, many RNA binding and modifying proteins, for example capping, splicing, processing factors, are recruited to the nascent transcripts. The mRNA exporter bind to RNA along with the adapter RNA binding proteins. The mRNA exporter acts as a Gtpa independent manner due to the structural unrelatedness to the karyophylline. However, like karyophylline, the mRNA exporter directly interacts with phenyl glycine rich repeats of FG nucleoporins and allows it to overcome the permeability barrier of nuclear pore complex, which is generated due to the FG nucleoporine meshwork. There are several general mRNA export receptors to transport mRNPs through the nuclear pore complex. Among these is MAX67 MTR2 complex and homologous metazoan TAP P15 complex, which is also known as NXF1, NXT1, are past studied export receptors. Although general Ren Gtp dependent protein export receptor CRM1 does not have any major role in export of mRNA export, but it has been observed that CRN1 can be involved in the nuclear export of several protoncogenes as well as cytokines, which contains AU-rich elements in their 3' UTRs. It is also been involved in nuclear export of number of unspliced and partially spliced viral mRNAs. The mRNA export receptor is targeted to different transcripts by export adapters that are typically mRNA binding proteins. The YRA1 is and can associate directly with general mRNA exporter. There are certain proteins that you call the SRS, which are serine arginine rich proteins, also function as adapters and regulate the multiple steps of mRNA metabolism, including mRNA export, their stability and translation. After splicing, several SR proteins remain bound to the splice transcript and then they are exported to the cytoplasm, where the dissociation of transcripts occur and SR proteins are re-imported again. SR proteins are basically evolutionarily conserved phosphoproteins and has been observed that cycle of phosphorylation and dephosphorylation of specific SR proteins. Basically, I'll give you the example that NPL3, it results in directionality during mRNA export in yeast. NPL3, when present in cytoplasm, gets phosphorylated by a protein that's called SKY1 kinase and stimulates its own import into the nucleus, where it can associate with nascent transcript in a transcription dependent manner. In the nucleus, NLP3 becomes dephosphorylated by nuclear phosphatase enzyme that's called GLC7, and then it interacts with the MAX67 MTR export receptors. After transport of mRNP into the cytoplasm, NLP3 rephosphorylated again, which destabilizes the interaction between NLP3 with the mRNA and the MAX67 complex. Now students, we talk about the regulation of nuclear transport through NPCs. During transport, there are certain proteins, for example, those binded to newly synthesized mRNA in the nucleus, contain both nuclear localization as well as nuclear export signals. These proteins frequently shuttle alternating between the nucleus and the cytoplasm. Their relative rates of their import and export determine the steady state localization of such shuttling proteins. If the rate of import is more than the rate of export, a protein will be located primarily in the nucleus, and vice versa for a protein located mainly in the cytosol. A number of shuttling proteins move insensate into and out of the nucleus, and in other cases the transport is strictly controlled. For example, regulatory proteins that control gene expression are only imported when they are required into the nucleus. Transport across nuclear pore complex is mostly controlled by regulation of NLS and export signal. Phosphorylation of amino acids in proximity of signal sequence can allow occurrence of these transport events. In other cases, binding of inhibitory protein to gene regulatory protein either anchor them into cytosol by interacting with organelle or mask their NLS, so that they cannot interact with nuclear import receptors. In such cases, transport across nucleus is possible only in the presence of appropriate stimulus that release cytosolic anchor or mask. Important example for such regulation is the latent gene regulatory protein that control the expression of proteins involved in cholesterol metabolism. After translation, this protein is stored in inactive form as transmembrane protein in the endoplasmic reticulum. Under distribute cholesterol levels, the proteins enter into the Golgi apparatus where specific proteases cleave it and release into the cytosol. Then this protein localize into the nucleus and control the expression of other genes acquired for cholesterol import and synthesis. Now students, we summarize the whole topic as nuclear pore complex are proteinous channels which are present in the nuclear envelope and are responsible for the active nuclear cytoplasmic transport. Nuclear proteins are imported from cytoplasm as their nuclear localization signal, that's we call the NLS, are recognized by soluble import receptors. Similarly, nuclear export signals, that's we call the NES, help in export of proteins from nucleus with the help of soluble export receptors. Presence of Rn Gtp in the cytoplasm and Rn Gtp in the nucleus is a key to control directions of transport. Export for nucleus is not only restricted to proteins only, and a wide variety of RNA molecules are also exported to cytoplasm for different cellular processes like translations. Export of small RNA like tRNA, microRNA, small nuclear snRNA follows the general pattern of involvement of exportin, that's we call the carophyrin family, and Rn GTP system. However, mRNA export does not directly depend on the Rn GTP system, and it's mechanistically different due to the usage of transport receptor that is unrelated to carophyrin. Different additional export factors like adapters and release factors are associated with the mRNA export receptors. Thank you.